Ouch! Some of those people are going to have some serious injuries in those epic fail videos. But have you ever stopped to wonder, how do those cuts and those injuries heal? How come if you get an epic injury on your knee, it doesn't last forever? The answer is mitosis, and that's what we're going to talk about today. Hope you have your green notes ready to go. All right, so let's talk about why cells divide. One of those important reasons that we just talked about seeing that video of those fails was for growth and for repair. Damaged cells are replaced by new cells. So whenever you're hurt or you get some skin scraped off your knee because you were trying to pull off a trick and it failed, well, those skin cells that you've lost are replaced because new cells grow in its place. Also, think about, how does an acorn turn into a huge oak tree? How does it grow? Growth in multicellular organisms like a huge oak tree happens because cell division makes new cells. This process of cell division starts with mitosis. Organisms grow because they make more cells. The cells don't grow in size. So when you saw the picture of the acorn turning into the oak tree, or here you have a picture of a newborn baby turning into an adolescent like you're growing because your body's making more cells, not because your cells get bigger. So technically, as a teenager, you have many more cells than you did as a baby because they have divided and created more. So why do cells divide again? Well, if you're only a single-celled organism, cells divide for reproduction. Think about our friend the amoeba. So we'll learn more about the amoeba soon if we haven't talked a lot about them yet. Every time an amoeba divides, it makes a whole new organism. So when you're a single-cell organism and you divide, and now there's two of you, there's two whole new organisms. But you are not unicellular, so your cells divide for growth and for repair. When cells divide and force those single-celled organisms to make brand new ones, that is called binary fission. So binary means two, so it fissions in half and there is binary. Now there's two new organisms. Cell division is very important for asexual reproduction, which only involves one parent. So that means that you have one cell and it splits in half and now there's two cells. That's called asexual reproduction, where it only takes one parent in order to make more than one offspring. However, every time cell division happens in asexual reproduction, the two offspring are identical to one another. So unless in that very rare occasion where we get a mistake in the DNA copying, remember what that's called? Yes, a mutation. So unless there's some sort of mutation, every time one cell divides and makes a new cell, the offspring will be identical, because we already talked about DNA replication and how that works. And so unless there's a miscopy, those two offspring will be identical. Sometimes asexual reproduction can happen in multicellular organisms. For example, this is called a hydra. A hydra is an aquatic organism where literally it buds. So a tiny bump appears on the parent and it actually buds and grows its own brand new offspring. So that is called budding, and it is a type of asexual reproduction. It creates an offspring genetically identical to itself by the budding process, which happens through mitosis, cell division. Genetic material, remember, is found in our cells. It is called DNA. Remember, what part is it found in? Yeah, that's right, the nucleus. Remember, that's some epic review right there. We should already know that the DNA is found in the nucleus. All right, on your green notes, there is a diagram of this cell. Please draw and label these parts. I know you've already seen a little bit of it before in your last notes, but we've got to review and know what our parts are. During most of a cell's life, the DNA and proteins are all a complex material called chromatin, so that bundled up stringy stuff. We saw that when we just spooled the DNA from the strawberry. Before cell division can happen, DNA is duplicated, just like we saw in our last set of notes where it makes an exact copy of itself, and compacted into these structures called chromosomes. Chromosomes are that X shape. Chromosomes are made of two identical chromatids. So remember we talked about pairs? So we have those chromatids. They're identical, held together in the center by a centromere. In the space on your notes, please draw and label all of these parts. That means the chromatid, each half, the centromere, and how DNA is coiled up into the chromatin. Got it? Great. Next, we're going to talk the stages of the cell cycle. There are three major stages of the cell cycle. The first stage being interphase. This is the part of the cell cycle when the cell is not dividing. It's when the cell grows. Yes, it does grow, but remember that's not why you grow. The cell grows and produces all of the organelles that it needs. Before a cell can divide, the DNA must be duplicated. Remember, that's where it unzips and then the matching base pairs come in to make identical copies of the DNA. The second stage of the cell cycle is mitosis. Remember, the cell spends most of its time in interphase. Then a short period of the cell's time is spent in mitosis. This is where the cell itself actually starts to divide, and it's where the nucleus starts to divide. So then you have two identical nuclei with two identical sets of DNA inside of each nucleus. So here's some more details from my favorite.
Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so I know that was a lot of information, but remember there are 3 stages of the cell cycle. Interphase, where most of the cell's life is spent, and then there's mitosis, which is when that nucleus is actually dividing and making 2 brand new sets of DNA. And then finally, stage 3 is cytokinesis, which is where the cell membrane pinches between the 2 new nuclei to make 2 brand new complete cells. Here's a cell sample of under a microscope, and you can see right there that cytokinesis just starting to happen, and you can barely see the line between the 2 cells where that cytokinesis is taking place and making 2 brand new cells. Please remember that the cycle begins when a cell is newly formed and ends when that newly formed cell is completely divided. This does not mean that the cell is dead. It has just completed a cycle. And that's all I have for you. Thank you so much for watching, and we'll be doing some mitosis activities in the next few days.